All right, my name is Alan Hurlbert. I'm a professor of biology at the University of North Carolina, Chapel Hill. And I'm going to start sharing my screen again with you and go back to this presentation here. So the things that I'm planning to cover today, first of all, I'm really excited to talk about this citizen science project called Caterpillars Count. So I'll be giving you a little bit of the background, how this project came to be, what sorts of questions it's designed to answer. I will show you a little bit, give you a little flavor for the kinds of things we've been able to learn from Caterpillars Count and that we hope to learn going into the future. And then most importantly, I hope to give you a sense of how you might be able to participate in this project and what participation actually involves in terms of time and effort and resources and all of those sorts of things. So as I mentioned, this URL here at the bottom might be a good one to bookmark for yourself. Okay, so I think probably most of us recognize that the Earth is undergoing various large-scale changes, whether that's climate change, urbanization, changes in land use, etc. And those changes are impacting potentially many aspects of biodiversity. One area that has received a lot of scrutiny in recent years is this idea of insect declines. The New York Times Magazine had this article a couple of years ago on the insect apocalypse. And there have been a number of studies from various parts of the globe suggesting that, a certain type of insect is declining, let's say flying insects in Germany or beetles in Puerto Rico, etc. So there are various reports. And of course, these reports are alarming for sure. What is unclear is just how widespread these sorts of insect declines are. And one reason it's unclear is that honestly, most parts of the globe, we don't have good insect monitoring data sets to begin with. Another reason it's unclear how widespread this problem might be is if in a location that does have an insect monitoring program in place, they monitor for 20 years and they find there's not really any big changes in insect biomass or diversity over that period, well, that kind of study is not likely to draw very big headlines. So this is actually a challenge that ecologists like myself face, is trying to generate data that might allow us to more systematically address this problem of insect declines at broad spatial scales in a way that's comparable from place to place. Now, another aspect of global change that might be impacting insects beyond simple declines is in terms of the seasonality or the phenology of insect activity. So hopefully, I don't know where the most widespread or the most northern participant on the Zoom call is today, but hopefully nobody is experiencing a picture like this one here. And most of us, at least certainly in central North Carolina, it very much looks like this middle picture outside my house, and hopefully for the rest of you, that will be coming soon enough. But we know that there's this seasonal transition, right? From winter to spring and summer, where the trees, deciduous trees, leaf out. And the timing of that leaf out may dictate the timing of a lot of insect herbivores that are using those leaves as a food resource, right? So for example, many caterpillars are soon emerging and will be taking advantage of that leaf out. But so one potential consequence of warming in particular is that the seasonal timing of spring, of plant leaf out, and therefore also of insect activity may be shifting a little bit earlier. And what we don't really know is to what extent that's actually happening, how variable those shifts are from place to place. So we kind of have these two underlying questions. The first one related to insect declines, where, by how much, and honestly, in the studies that have documented insect declines, we don't really have a great understanding of what might be driving those declines. Well, we do have a good understanding of maybe a list of, you know, six to 10 potential drivers of declines, but in terms of actually figuring out which of those drivers are most important, I think it requires more data from more places to understand, oh, insects are declining here, but not there. They're declining more here, but less so over there, et cetera. And then we can start to compare across these sites and try to get at some of the differences in the drivers across those sites. And then the second one is this question about shifting in the seasonal timing. So insects, and understanding why insects might be changing in these ways in response to changes in the environment. It's important for understanding the ecology of insects themselves and what's happening to our biodiversity. But a confession is that I'm not an insect ecologist. I'm actually a bird ecologist. And so my concern actually stems from the fact that there are many migratory birds like this indigo bunting here. And this animation is based on citizen science observations from the eBird project run by the Cornell Lab of Ornithology. And these millions of bird observations give us this picture of our animation. Oh, there it goes. The animation is still going there. It gives us a sense. Actually, we know very well in any given year exactly when indigo buntings start, migrating northward, when they arrive on their breeding grounds, when they start migrating back south again. And of course, the reason these migratory songbirds in particular are migrating north to begin with is to take advantage of this huge pulse of insects that are suddenly available in large quantities in the northern hemisphere summer. So if insects are declining, or if insects are shifting the timing of their activity, that has the potential to have dramatic consequences for a lot of other organisms like birds that may depend on those insects. So while birds are one group where citizen science has been extremely successful in harnessing the power of lots of eyes of observers all around the world, we've had less concerted efforts to better understand what's happening with the insects. And that's really where this project, Caterpillars Count, comes in and tries to fill a gap. So some of the aspects that I'll be talking to you about today are, how these, the basic unit of participation is looking at a branch and describing, okay, are there a bunch of insects on this branch or not? Which insects are there? How many of them? We have mobile apps. I'll talk a little bit about the free mobile apps and web-based data submission for submitting your observations, resources for learning to recognize some of these different types of arthropod groups. Our website has built various data visualization tools to explore the data, not only the data that you yourself might collect, but data that anyone in the project has collected across the globe. As I'll get into, this project, I think, is especially well suited to nature centers, school groups, master naturalist groups, etc. But equally well, people that are particularly interested in just understanding the biodiversity of their own backyard might be able to set up a site and monitor that site as well. I don't know if we have any formal or informal educators on the call today, but we have a variety of learning activities. Some of these are ones that we have designed ourselves, but we also just link to a wide range of learning activities related to insects, climate change, food webs, phenology, those sorts of things. So I'll point a little bit towards some of those resources at the end of the webinar today. So up to this point, we have engaged over 150 sites across North America, primarily Eastern North America to date, but there's no, we welcome participation from, we would love to see this map filled in, right? We would love to see, all 50 states and all Canadian provinces participate in this project, because ultimately we do hope to get a continental understanding of how insects and arthropods as bird food, but also in their own right, how they vary across the map from year to year, from tree species to tree species, et cetera. So far we've engaged at least, actually the number is higher now, but more than 1,500 people have actually submitted data. Far more people have been involved in that data collection. So far, we've had 68,000 branches surveyed and over 160,000 arthropods observed. I guess I should say that this project actually started in 2014 was sort of the pilot year. In 2015, we had a handful of sites, primarily North Carolina. But since that time, we've been growing and expanding. So we're very excited to get participation from by new folks at new locations throughout the globe. I actually see in the chat some members of the Fairfax chapter of the Virginia Master Naturalists have participated since 2018, which has been fantastic. And thanks for joining in on the webinar. So I wanted to start to just give you a flavor of some of the things that we've actually learned, and also that sort of points towards things we still have yet to learn or better understand. But one of the things we did at the outset of this project was we wanted to just verify that the data collected by the general public is actually reliable and useful data. And so what we did is at one of our nature centers here in North Carolina, we compared the data collected by citizen scientists, volunteer participants, with the data collected by trained scientists. And so across a number of axes, we found that the citizen science data was great. It was very reliable. This is just showing the proportional breakdown of different arthropod groups found on branches. This middle panel is illustrating that the density estimates, that is the average number of arthropods of a given type. So the color of these symbols refers to the different arthropod groups. And the value, its position along this x-axis here is the estimate obtained by the trained scientists. And the value on the y-axis is the density estimate obtained by those volunteer citizen scientists. And any data points lying exactly on this black one-to-one line here would indicate that both groups had the exact same estimate. Now, a lot of these points, they don't lie quite on the line, and yet the line runs through all of those points. It's a very good approximation. So on average, both sets of groups were making very similar estimates in terms of which arthropod types were more abundant or less abundant, et cetera. I'll get into this difference about survey method a little bit later on, this visual and deep sheet surveys. And then this last panel on the right here is illustrating how the two groups arrived at very similar patterns in terms of the phenology, or that seasonal timing, right? So both groups identified a peak, an initial peak in sort of mid to late June in caterpillars, for example, with a little dip. I think there was actually a week, this gray line, there's just a missing data point in here. So it's connecting directly from the end of June to mid-July. But so these results, first of all, made us feel pretty confident that the data that these volunteers collect can be used to estimate these various quantities. One of the things that we've realized is from this project is that at any given location, one might conduct surveys for these insects and arthropods on a number of different kinds of tree species. And that host plant identity actually really determines or explains a lot of potential variation in why there are more bugs in some places than other places, or on some trees versus other trees. So this is just an example. This is from a site, again, right near me in Chapel Hill, North Carolina, the North Carolina Botanical Garden. And you can see we have a variety of tree species arranged at that site, and the variety of different types of arthropods that have been observed per branch survey on those different tree species. So if we were to care about caterpillars in particular, for example, it looks like red oak has this big, fat, pinkish magenta bar here relative to, say, sugar maple. So we can make these comparisons across. Now, just because there may be attributes of red oak that are really good for supporting caterpillars and make them much better than sugar maple, but it's not obvious that that difference necessarily is going to be maintained at every location. So host plant quality and relative host plant quality may vary a lot from place to place. And so that's part of what caterpillar count data can help address. I mentioned that this project has been going on since 2014 or 2015 in some locations. So again, this is for the North Carolina Botanical Garden. And these surveys, because we've been surveying the exact same branches for every year over this time span, it's allowing us to evaluate trends. And so this is just an example. This is taken from the data visualization tool on our website, but this is showing 4 different arthropod groups, and all 4 of these groups appear to be exhibiting weak declines. Now, this is just a 7-year time series so far, so we wouldn't want to over extrapolate, and potentially to some degree, maybe 2015 happened to be an unusual year, and abundances happened to be higher than normal in that year. If you look at just the rest of the time series, maybe it's not been such a steep decline. But this is one of the things that we're really concerned about, right? And so we hope that for those who are interested in participating in this project, that one of your motivations might be better understanding the insect biodiversity that's around you in natural areas or in your yard, but also generating this baseline data so you can better evaluate the extent to which things are changing over time. In many respects, we expect insects and arthropods to be the canaries in the coal mine for evaluating our ecosystem health, right? So many other organisms depend on those insects. And so if we can detect those changes in the insects, we maybe can better, we can better address some conservation concerns at sort of an entire ecosystem level. Now, I'll mention iNaturalist a little bit more later on, but I'm sure many people on this webinar have heard of the project iNaturalist, which is a fantastic project. All it takes to participate in iNaturalist is, a phone or a camera. You snap a photo of some cool thing that you observe, and other folks can help you identify it if you don't know what it is. And it contributes to a large biodiversity database where it generates a better understanding of where different species occur and when they tend to be active or flowering or whatever. And iNaturalist is wonderful, and there's plenty of people that are participating in it, and I hope it continues to grow. The one place where iNaturalist falls short is in its ability to generate estimates of abundance or density, because the problem is that inherently we only have the record of when someone saw something. We don't know how much people went out and didn't observe a thing. And that negative data, that absence of observation, is really critical to coming up with comparable estimates of density or abundance that we can better compare from year to year, place to place. So that is one of the sort of gaps that this project Caterpillar's Count is ideally suited to kind of fill. Okay, so I want to talk a little bit about how what it looks like to set up a Caterpillar's Count site, what's involved in that. And inherently, it is designed to be very flexible because we recognize that different individuals or organizations will have different capacity and people power to devote to a citizen science project. And so we want to kind of build in that flexibility, allow places that have greater capacity to do more, places that have less capacity to do less, all within this framework. But I'll first just talk about a suggested setup. Let's say if you have a small group of interested people, maybe it's people, it could be a nature center which runs citizen science walks every week, or a group of master naturalists. It could be a school group, but some group where you have the ability potentially to have 4 or more people that can kind of divide and conquer the effort. So this would be a suggestion for a group of that size. Basically, a Caterpillar's Count site involves a set of survey trees or survey branches. I'm going to use trees and branches kind of interchangeably here. Typically, every survey branch is going to be on a different individual tree. And so what we recommend is setting up a site with at least 30 survey trees. Some of our sites have 40, 60, even 100 trees. That could be a lot of time and effort. We don't expect everybody to have the capacity to survey 100 trees. But 30 trees, based on our preliminary analyses, gives us estimates of arthropod abundance that we feel pretty confident in. Below 30, and insects can be very patchily distributed across the environment, right? You might look at 5 or 10 branches and really not see very much at all, and then you look at that 11th branch, and suddenly there's all this stuff. And because there's this inherent noise to the distribution and abundance of insects in the environment, that's why we kind of need a large-ish sample size to feel good about our estimates. So what does it mean to have 30 survey trees? Well, these survey trees are arranged in groups of 5. So that's what's being represented in this diagram here. And we call these groups of 5 a survey circle. A survey circle has 5 survey branches. Typically, there's one central branch, and you might pick the location or identity of this central branch based purely on the fact that, okay, I have a sense of where I want to do some surveys. Here's some good vegetation to monitor, and here's an area that has that vegetation that I can access. So maybe I'm going to pick a tree centrally within this little region. But then, and so that might be a subjective choice. And then these 4 satellite trees around that central one, ideally are chosen in a more systematic or standardized way, which is basically, okay, walk 5 meters in each cardinal direction, for example, and then just pick the closest tree that you come across. And here there's plenty of flexibility, because often you're limited, depending on where you are, you may be completely limited in how much vegetation you have to choose from. And so, maybe you walk 5 meters in one direction, but there's nothing, there's no trees in that direction. So it's fine if you end up, if they end up a little bit more clumped looking or something like that. But the reason that we encourage this, this sort of standardized tree branch selection is because we want your tree branches overall to be kind of representative and not to be biased in terms of what tree species are selected. So obviously you're limited by being restricted to where there's vegetation. But then assuming you're in an area of vegetation, then we have these four out of the 5 in each of these groups is sort of chosen in this standardized fashion. I should mention at this point, since I haven't, well, I've mentioned tree and branch. This project very much is focused on woody vegetation. So trees and shrubs. And of course, as you'll see in a moment, as I demonstrate some of the survey methods, it's very much limited to tree branches that you can actually access, right, that are at eye level. So if you have trees where the branches don't start for 20 or 30 feet, obviously you're not going to be able to survey those branches. So when I say available vegetation, I'm really talking about vegetation down, woody vegetation at ground level that's representative of your site. I'll get into it a little bit more in a moment, but if you allow about 5 minutes per survey, 30 survey trees would take 2.5 person hours. So again, this is very different from participating in iNaturalist, right? Where, oh, I see a cool thing, I take a photo, the whole thing took about 15 seconds and I'm done. So that's very easy to get large volumes of this very granular data about this species was observed in this location on this date. But again, if we want to get estimates of density or abundance, it requires a little bit more effort. But this 2.5 person hours, as I mentioned, if you have a group of 5 people, then that's a group of 5 people just spending half an hour outside together, right? So that, again, if you have people to divide this work divide up the surveying, then it can go quite quickly. I have on here weekly monitoring from May to July. Now, when you create a site, and I'll show you how to do that in just a moment, you can create a site with as few as 10 survey trees, as many as you want in multiples of 5. And you can monitor just one time, or you can monitor, 3 times a week, all year round, as long as there are leaves on the tree. All of that part, so we don't dictate that. You don't have to seek permission for, well, I only want to participate at this level, etc. Anyone can create a site, don't need permission to do that, and you can sample as many trees as you would like at whatever frequency you would like. This is our suggestion, and this weekly monitoring for May to July, the reason that's up here is our recommendation. That's the window of time that's especially critical if we want to evaluate what's going on with that seasonal timing of arthropod availability relative to the timing of migratory birds and their reproduction. That's the window where they're migrating to breeding locations. They have chicks in the nest whose fledging success in part depends on the timing and the abundance of insect bird food during that window. So those are our suggestions. Now, for those of you who are maybe not affiliated with any particular organization, but you really like bugs, you like learning about what's going on around you in your backyard, you're really interested in phenology, maybe you participate in some national phenology network projects or something like that. Absolutely, feel free to set up a site in your own backyard or in your park. Maybe later on you meet some friends and decide to work together. But if you want to just do something by yourself, sure, you can set up a site. Again, the minimum number of survey trees to commit to is 10 survey trees or 10 survey branches. So 2 of these groups of 5 survey circles. And again, at 5 minutes of survey, that's less than an hour. And again, if you don't have lots of options for choosing that vegetation, the spatial layout does not necessarily have to look like this perfect little X or cross or plus sign. But it can be, if you just have a backyard with trees and shrubs around bordering the back, those 10 branches can just be in a line. So it all is flexible and based around what you have to work with. So here's just a particular example to make it a little bit more concrete. Again, this is the North Carolina Botanical Garden, which I've shown a few examples of data from before. And at the Botanical Garden here, we have 40 survey branches. So that is 8 of these red dots, of each of which is a group of 5 branches, right? So with the branches 5 meters apart from that center branch. And so these, although we have a traditional sort of display garden, a lot of herbaceous plants and stuff like that in the display garden, outside of the display garden, we have a nice natural wooded area, a trail running through some woods with a stream. And so we've identified various branches. We've kind of distributed these 8 survey circles along this trail. And so we can, there's a group of us each year that walks around and hits all 40 of those survey branches. So again, depending on your own natural area, you can kind of spread these out as close or as far apart. I mean, the only real requirement, right, given that a group or a survey circle, these things are 5 meters apart. So you would want the next survey circle, the edge of the next survey circle to be at least 5 meters away from the edge of another survey circle. But other than that, you can pack them in tight if you are limited in space, or you can spread them out, over a mile or more, just depending on what trails or general areas make sense in your circumstances. Okay, so I mentioned there's no permission required to create a site. There's a website. All that's required basically is to go to our website, caterpillarscount.unc.edu. I'll demonstrate the website shortly. But you do need to create a personal account first, create your own Caterpillars Count account. And then having created a personal account, you get the option to create a new site. And here's all the information that's required. You just have to come up with a descriptive site name. If you're part of an organization, you can optionally put that in here. You can describe briefly sort of the nature of the natural area or something like that, the area that's being surveyed. And then you'll choose, okay, how many plants or survey branches are you going to do? And at a minimum, as I mentioned, it will minimum will be 10, and then you can increase that in increments of 5 branches or 5 plants, up to as many as you would like. And then the final piece that you have to provide to create a site is creating a site password. And so we have this site password in place just to kind of prevent spamming of fake data or things like that. So the idea would be anybody who's actually contributing data from this site will have to be told at some point, oh, our site password is, I heart caterpillars, or whatever it is you choose. So this type of site password is, this probably is best chosen to be something easily memorable, something you can verbally share with volunteers. They have to put it in, if they're using, for example, our mobile app, which I'll demonstrate in a moment. It's something they enter one time, and then the app will remember, oh, this person has permission to submit data from the site, and then they're good to go. And of course, if you forget the site password, you can reset it at any point. So that's it. You fill out just those little bits of information. Oh, it's obvious from the diagram here, you do have to show where it is. So you would zoom in on the map as close as possible, place a pin for where your site is. But that's it. And like I said, you don't require permission from me. You can go and do that this afternoon if you're so inclined, and your site will be up and running. Now, let me just gonna break out of here for a moment and actually show a little bit more of the tools for once you've created your site. So here's the website, caterpillarscount.unc.edu. And once you have created a site, then you can manage it. Here's the Create New Site button. Here you can manage a site. So presumably you'll just have access to one particular site. I am Caterpillars Count deity, so I see all the sites here. but let's pick Acadia National Park, let's say, up in Maine. So they've been a great participant for several years now. So as soon as I've created a site, let's say I created 24 survey circles, which means 20 survey branches at this particular site in Acadia National Park. What happens is that our website automatically creates for you a set of unique 3-letter codes. So here are some examples here. And of course, we have no idea what plant species you're going to place these, or you're going to select for your actual branches. But once you have walked around your site and settled on branches, if you're able to, then you can enter this, the plant species yourself. So you can see here, circle 1 has 5 branches: red, blue, green, yellow, and purple. Each of those has a unique code. You can go around and then you can figure out which branches you want to use for those. You can fill this in using either scientific name or common name. And so, and you can actually do that. You can walk around and do this on your phone. If you have web access, you can fill in the plant species as you're walking around. So once you've filled those names in, then, oops, sorry, let's go back and select that. Our website automatically creates these tags that you can print out. So these are the unique 3-letter codes. It's telling you, oh yeah, this particular code is for survey circle 1. You filled in that it's American mountain ash, so that species identification gets added to the tag. And then you can print these out. You can cut these tags out. You can the back is just a little QR code that so when you hang these tags from your survey branches, you can find them more easily. Visitors can use these just to learn how to identify, plant species, but also they can learn about the website by scanning the QR code on the back of the tag. So you can laminate these. Often we do the poor man's version of lamination. The size of these is designed to fit inside just standard 2.5-inch wide packing tape, right? So you can double fold it inside packing tape, punch a hole, hang it with a twist tie or something like that from your branches. When you first create the site, of course, that plant species will just say NA. So you probably don't want to print them out until after you've filled out that information. So let's see, sorry. I'll go back to, oops. This page. I guess the one other thing, as long as we're here, I'll just point out that so whoever creates the site is, of course, the default site owner. And that site owner, I'll maybe talk a little bit more later, has the ability to, manage all the data that comes into that site, to edit the site details, to add information about the plant species and all of that. If you would like to share those responsibilities with someone else, all they need to do is create their own Caterpillar's account, and then you can add them by putting whatever email they use to sign up here, and then they'll show up, and you can give those people the additional power to add yet more site managers, etc. This is a really important tool if you have staff turnover, right? Let's say you have one person sets up the site, but then they leave their position after 2 or 3 years, and you want to make sure someone else, the next person in that position, has the ability to continue that management role. Then you can add them with this Oversee Managers tab here. Okay, so let's talk a little bit more about what these surveys actually entail. And I've already alluded to the fact that there are 2 different survey methods. And so I'll demonstrate both of those today. they each kind of have their pros and cons. I guess the first one is the visual survey. The visual survey, the main pro is that you don't need any special equipment. You can show up. Now, of course, you need a way to record the data. That might be paper data sheets that you could print out from our website, or it could be ideally using our mobile app. And the reason I say ideally using the mobile app is because it has some, well, it saves you time in that you don't have to spend time writing it down, in one moment, and then later on, someone, that same person or someone else, has the burden of having to type and enter all of that at the website later on. But the other reason is that the mobile app has some built-in checks. Like if you happen to write down a number, try to enter a number in the moment, that would be really, unreasonable or unusual, then the app will actually prompt you or make you, double check things like that, right? So that's why we do like people to use the app when possible. But in any case, assuming you're using paper data sheets or the app, you don't need anything else. You can show up to your site and you can go to a branch and you just manually inspect that branch over the course of a visual survey. And I'm going to stop sharing again and actually unblur my background to share with you this nice little fake potted plant. If I'm being honest, it is a fake plant, but it has on it a little unique three-letter branch code that was produced from the website here, right? So again, I've set up which branches I'm planning on surveying in advance. So now I know, oh yeah, this is BYT. I'm going to come up to this branch and I'm going to do a visual survey. So what does a visual survey really mean? A visual survey is a survey of an area of 50 leaves, 50 leaves, and the associated twigs, petioles, and stems associated with that set of 50 leaves. And that's going to be a contiguous set. So I wouldn't examine a few leaves here and then a few leaves way over here and a few leaves over here, right? So some continuous area of 50 leaves. And the reason we set it to be 50 leaves is just to we need it to be standardized in some way to help us compare from branch to branch. Because of course, some branches inherently have more leaves than others, right? So if we just said, oh yeah, go survey any old branch from a tree, there'd be tremendous variation from plant to plant and from site to site in how many leaves people were surveying. So we're interested in all of the arthropods on either the top or the upper or underside of the surface of these leaves, again, that are on the twigs. So it's going to require, you can't just sit back completely. You're going to have to get in here. You're going to have to examine manually, like, maybe flip the leaves over. A lot of this depends on the morphology or the branch structure. Some branches, you can like hold one stem of the branch and examine all 50 leaves, the underside, and all 50 leaves all at once. And then other tree species, the leaves are very overlapping and you have to almost go through leaf by leaf. But so you want to inspect everything. And the challenge here, of course, is that the things that you're documenting have undergone millions of years of evolution to not be detected by large vertebrates that might do them harm, right? So many bugs and critters are quite cryptic in their coloration and position, etc. So we have to, spend some pretty careful time looking at all of this. Now, if I were to go leaf by leaf, I know I need to look at 50, right? So let's say, okay, I'm looking at leaf 1, 2, 3, 4. Oh, look, there's this cool little bug that I drew with a Sharpie. So let's say that's a real bug, and I want to note that. I'm going to start entering some information about that, which I'll show you in a moment. but now I've forgotten how many leaves I've been counting up to that point. So what I usually do is I actually count out the area of 50 leaves before I even start worrying about the bugs. And I try to count like, oh yeah, it's this part of the branch up to some particular landmark, right? So literally, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 50 takes me, right to this particular fork. And then if I remember that landmark, I don't have to worry about counting those leaves again. So the visual survey, you are visually inspecting all of that. Because you have to be careful, because things are cryptic, right? And you're trying to be careful and inspect all the surfaces. We estimate on average, it takes about 5 minutes to do one of those. The alternative survey approach is called the beat sheet. And the beat sheet, this is an example. So there are beat sheets you can buy, but we have instructions for building a really simple collapsible beat sheet on our website. This thing. So this thing is designed so it easily folds up. I have a couple of pockets that these bottom. So this is just basically buying a 10-foot piece of like cheap pine molding, cutting it into 4 pieces of 2.5 feet each, and then this is a 2 by 2 foot sheet, and it's really just stapled on to 2 of the sides. So there's instructions for how to build one of these. You could, if you already have like an actual insect beat sheet, you can use that as well. But the idea of a beat sheet is you hold it under the survey branch and you get a stick, and I don't have an actual stick in my hand, but imagine I'm holding a little stick and I whack that branch 10 times. And for the most part, any arthropods that were on that branch are going to fall down into this beat sheet. And now we don't have to worry quite so much about that camouflage, right? Because they become much more readily observable on the beat sheet. So the beat sheet is a bit faster. It's great for sharing your finds with other people, because now that they're in the beat sheet, you can kind of show it around to people in a group. If you're doing this exercise with younger participants, kids, right? It's much fun. It's much more fun for kids, both to do the beat sheet, but also just to see the bugs on a beat sheet, etc. But you need the beat sheet, and you may need, to have several beat sheets, and you may need to arrange a way to get those beat sheets to the volunteers who are doing the surveys and things like that, right? So, like I said, pros and cons, and whichever one you find most useful for your scenario, it's all good with us. We just ask that you choose one method and that you just stick with that method. There are slight differences in the likelihood of seeing different types of bugs depending on which method. Happy to talk more about that later on. But as long as you're consistent, that's the most important thing. Okay, let me actually, I'm going to go back to sharing my screen, and I want to show you, well, I guess we have it in this presentation still. So here's what the actual mobile app looks like. So once you sign in with your personal account, you'll go to some branch. This actually, we happen to have, you'll see a document if you go to our website resources for practicing with the mobile app. So this is a site, a branch that's designed for you to just practice with. So you can use the app and actually submit data to this practice site, but it won't, gum up our database with fake data. So we have these various bugs that are kind of photoshopped on here, right? So a branch has a unique 3-letter code. So that's the very first thing you enter. You enter, oh, I'm at branch EJR. And as soon as you type those 3 letters in, the app knows, oh, you mean you're at example site, survey circle 1, it's an American beach or whatever. So based on what you've entered before, when you've created the site, it will know just from those 3 letters where you are. The very first time you submit data from a site, you have to submit that site password. So remember, this is the password. Whoever creates the site is going to come up with something easy to remember to share with volunteers. They're going to enter it the first time, and then every time thereafter, this will not even show up on the app. They don't have to worry about it. Automatically, it records the date and time. You select whether you're doing a visual or a beat sheet survey. You can record general notes about, that maybe there was a big storm or something like that. The leaves are wet. There's a little checkbox to record that sort of thing. But then once you say, yeah, I'm doing a survey, the main question is, well, did you see any bugs? Did you find any arthropods? And if you select yes, then you get a drop down. This is just the top of that drop down menu. So something I haven't talked very much about yet is kind of at what level we're expecting people to identify things. Well, for this to be a successful scientific citizen science project, we certainly do not expect everyone to be insect taxonomists or systematists. I myself am not able to identify almost the vast majority of things to family, let alone genus or species, right? But what we do expect everyone to be able to learn is how to distinguish groups more or less at the level of order. So order really, examples of orders would be distinguishing, say, beetles from bees and wasps, or aphids, caterpillars, flies, spiders, right? So pretty coarse level. In a few cases, we have identification below that level. So for example, ants are within the order that includes bees and wasps, but they're so obviously distinguishable, we have them as a separate group. Aphids and psyllids are a suborder within the order that includes leafhoppers and other true bugs. So that's the level. And so you would look at your branch and you decide, oh, do I have any of these things? And if the answer is yes, then you go ahead and you enter the information. So in this case, on this survey, let's say we say, oh, we saw 4 ants that were 3 millimeters long. So that's the other piece, is not just what did you see, but how big was it? And of course, this is pretty important for if we want to translate this information ultimately into arthropod biomass. We can use these length estimates to come up with biomass estimates later on. So we want to record the difference between an ant that's 3 millimeters versus an ant that's 10 millimeters long, right? So, okay, we can make separate entries if it's the same type of bug, but a different size. So 4 ants of 3 millimeters, 1 ant of 10 millimeters, 1 caterpillar of 35 millimeters. And oh, I saw something else. You can click the add more arthropods. You can keep adding until you've recorded all the things you came across on your branch. At that point, you continue. And then the last set of information we need is regarding the branch and the leaves that you examined. So I mentioned beforehand that a visual survey consists of surveying exactly 50 leaves. If you selected visual survey, this number of leaves is going to automatically fill into 50, and you're not going to be able to edit it because you were instructed to survey 50 leaves that shouldn't be editable. If you do a beat sheet survey, well, the number of leaves that are hovering over that beat sheet when you whack it, that actually is just going to vary from branch to branch. So we ask people to estimate, okay, just how many leaves do you estimate were above the branch when you were above the sheet, sorry, when you beat it. And then secondarily, we ask people to estimate the length of the blade of the leaf. And again, we will use that information later on, depending on the plant species. If we know the length was 8 centimeters, we can convert that to sort of leaf area. And then we can multiply that times how many leaves you surveyed to get a sort of a total estimate of leaf area surveyed. This plant species, that will populate automatically, assuming you have entered that, whoever the site owner is, filled in the plant species of each of those unique branch codes. The person doing the survey doesn't need to enter that information. That will just fill in automatically. And then the very last piece of information we ask people to assess is how chewed up are those leaves. And so that's what's called herbivory. In many cases, you might look at a branch and you might not find any caterpillars, but you might see lots of evidence that caterpillars had been there because the leaves are extremely munched on. So we have a few sets of, we have basically these categorical scores of herbivory. And there's some documents that help people to get better at estimating, knowing how to distinguish, let's say, 5% or less from more than 5%, 6% or more, for example. Okay, let's see. I noticed a couple questions in the chat, so let me get to them. First, is there a website or course for identifying caterpillar species? There are some identification resources for identifying caterpillar species in particular. The best resource I can recommend is this book by David Wagner called Caterpillars of Eastern North America. But caterpillar diversity is huge in North America, and this book covers only a fraction of it. So it's a very good introduction to kind of the different groups, the different families and subfamilies and common genera. But it's, it actually getting really good with caterpillar identification probably requires delving into, finding various scientific keys and things like that that are available online as well. We have a number of other resources for identification on our website as well. But one thing I forgot to mention with the app, which I think is very cool, is that optionally you can take a photo of a bug that you see on your survey branch over the course of your survey. And that if you do that with our Caterpillars Count app, the photo automatically goes to iNaturalist, to the Caterpillars Count iNaturalist account. And that is where other experts might view that photo, and that's where, in many cases, we might get a species level ID. Even if you yourself didn't know what species it was, you just said caterpillar, you took a photo, the caterpillar photo went to iNaturalist. Someone else is able to tell you, oh yeah, that's a sycamore tussock caterpillar, or a whatever it might be. So we strongly encourage people to use that feature, to take photos, especially of caterpillars, because caterpillars, the reason we call the project Caterpillars Count, even though we survey spiders, someone asked a question about spiders. Yes, spiders, beetles, fly, whatever you see, snails, sometimes you come across snails. People have come across tree frogs on their survey branches. There's an other category when you select what you came across. So you can record anything. But especially any kind of arthropod, we definitely care about. But caterpillars really do have a special place with respect to serving as bird food and bird food for hungry nestlings. Many songbirds, even if the adults tend to more specialize on seeds, like many sparrows, they feed their young caterpillars more often than not. And so caterpillars really do count in that respect. And of course, many people are also especially interested in butterflies and moths. And so having this understanding of whether caterpillars are declining in abundance or shifting their seasonal timing is also going to help us better understand the abundance and seasonal timing of butterflies and moths as well. Yeah, the book was Caterpillars of Eastern North America by David Wagner. And it's a really nice, well done book. Let's see. Someone else asked, does the time of day matter for doing the surveys? It does not matter from the project perspective, as long as you try to keep that time of day consistent from week to week, year to year, that you tend to have, you yourself are doing these surveys, or you're having volunteers doing those surveys roughly the same time. Insect activity does vary over the course of the day, and that varies maybe a little bit by type of insect group, like what time of day different insects are sort of most active, etc. So I think that usually especially at a nature center or for a group of people, the biggest driver of that decision is going to be the ease of logistics and what time makes the most sense for people to reliably, meet on a frequent basis. But so yeah, so that is pretty flexible from our side of things. It's whatever works best for you. Okay, let's see. And then yes, this recording will be available. I will email this the link to the recording out later in the week. Okay, let me I also wanted to show, I just wanted to demonstrate a few more resources and then kind of make it wide open for other questions that are occurring to folks. So this virtual survey that's being advertised here can just give you, you can do this without even creating a Caterpillars Count account. And it can give you a sense of really what is being asked of you. So it's on our website, the Learn tab, and then this virtual survey game. And I'm going to make it full screen here and hit play. And the first level is quite easy in the sense that it's this branch with a white background. So of course, participating in Caterpillars Count requires, first of all, finding bugs on branches. Are there bugs on branches? If so, oh yeah, can you find them? So I'm going to click on this guy. Then here's the drop down that comes up. And this is exactly what the app looks like. So I'm going to look at this guy and I'm going to say, oh yeah, I recognize this little triangle shape and the pointy shoulders here. And I know that's a true bug. Now I need to estimate its head and body length. So not counting antennae, not counting legs, just from here to here. And I have this ruler up here and I'm just visually comparing. Now in practice in the field, when I'm doing real surveys, I always suggest that people learn the exact length of their thumbnail and their pinky nail in millimeters, because most bugs are going to be in that range. And it gives you a ruler, a benchmark ruler that you will always have with you. But here I'm comparing to this ruler up above. I'm going to go ahead and estimate 8 millimeters here. And oh, I underestimated it, right? So it's actually 10 millimeters, but I successfully identified it. So that's great. And I'm going to pretend like I don't see that moth there and say, oh, I'm done. And it shows me, oh, wait, I missed one. And then the next branches get a little bit trickier, right? So now it's a little bit harder to find these things. And I still have to identify these things. This is a cool looking caterpillar. I've got it now in my mind. I need to stretch this guy out so that he's straight. And in doing that, I'm going to go ahead and guess 15. And,
Alan, we can't see your screen.
Oh, thank you so much for telling me. That is crazy. Thank you. Sorry about that. I was going on for a very long time. Sorry for those who maybe tried to tell me that. Oh yeah, there's all the chats. Now I'm getting all those chats. Sorry about that. Okay, so I'm in this virtual survey. Here's what I was talking about, which is you're seeing this virtual branch and there's bugs on it, which I can go around and look for. So it's just practicing doing one of these surveys. So when I click on the bug, it brings up this drop-down menu. I can say, oh yeah, that's a moth. I'm estimating how long, what's the head to body length. And so in this case, the moth is longest from wingtip to wingtip, but I'm estimating the head to body length, which is this length here. So I'm going to go ahead and estimate 7 millimeters. So this is the feedback I get. Oh, I successfully identified it correctly. I got the length correctly. And so it lets me just go on, and there's a time limit. So this game is a fun way to just practice all three of those things: finding bugs, identifying them, and estimating their length in millimeters. Okay. And there it's showing me the ones I missed. Let's see. I see another question in the chat, which is, should we choose broadleaf trees rather than needle leaf trees? And that is a great question. Let me go to, so you are welcome to choose whatever trees are representative. You can, so up till last year, we focused exclusively on broadleaf trees, in part because our methodology of, like, say, defining how many leaves or examining an area of 50 leaves, it didn't really translate to coniferous or needle trees. But we actually have another, it's the same survey methods. It's a visual survey or a beat sheet survey. But instead of dealing in number of leaves, we deal in linear branch length sampled. So this is the kind of thing that you would record when you, for example, if you did a beat sheet, we kind of ask you, and it might take a little bit of practice estimating this, but okay, just how many centimeters worth of coniferous branches were hovering over that beat sheet? That's what you would record instead of the number of leaves sampled. But you absolutely can do beat sheets on these, and you can do a visual survey. But when you do a visual survey, you need to come up with what you're going to use as kind of a standard estimate. And of course, this is the tricky part, is like, how do you train people? How do you yourself get comfortable saying, okay, I feel good that I examined basically 100 centimeters worth of branch or 300 centimeters. So we're still kind of in the newish phases of this, but we absolutely are trying to build in that flexibility to allow people to sample. And so there's this special instructions on our website for doing conifer surveys. That is a great question. Okay, let me come back to this presentation here. I guess the last couple of things I want to show off really briefly. So one is, if you go to Explore, we have a Maps and Graphs page. And the top of the Maps and Graphs page is kind of just like a caterpillar leaderboard for the year. That's more just for fun to see which sites, are seeing the most caterpillars. But for actually exploring and visualizing the data, so this map here shows, you can see these dots illustrate all of the places where we have caterpillars count sites. And they're actually shaded at the moment from dark green to light green based on the percent of surveys that had caterpillars. And so you can get a sense, okay, most of these sites are dark green, so they're down on the lower end. There's a few sites, if I hover here, so this site here had 50% of its surveys had a caterpillar on it. Whereas it's much more common for sites to have maybe in the single digits, maybe 5% of survey branches might be really common. And that's also another reason to do more survey branches, to have more survey branches at your site. You're increasing the likelihood of coming across cool critters. So that's what's being visualized at the moment, but importantly, we can change what's being visualized across that map. We can visualize spiders, for example. And let's say instead of spider, the percent of surveys, we want to visualize the biomass per survey. Remember, because you all help estimate the length of the spiders you're observing, we in the background convert your length into a biomass estimate. So we can calculate that biomass for you. If we wanted, we could zero in on a particular survey methodology. We could say, oh, we only care about large spiders. Let's say spiders that were 10 millimeters or larger. Or only spiders occurring on a certain tree species or in a certain year or month. So we can select all of these things, and that'll change what's being displayed in the map below after it processes that query. And you can see, oh, it looks like Riverbank Zoo in South Carolina, really good for spiders. And that 173, that's milligrams of dry mass per survey, I guess would be the units there, compared to most other places, which are quite a bit lower. So you can visualize that survey data. You can download that data to play with on your own if you like. But then if you want to explore the data from your particular site, so I'm going to zoom into North Carolina here. Here's the Botanical Garden. If you click on that site, you can get an overall summary, how many surveys have been done at this site, how many caterpillars or total arthropods have been found, what's the leaderboard look like at your site for, how many caterpillars people have found, etc. Let's see, out of all time, oh my gosh, I'm dropping off the top 10. But so you can see, right? So different folks have seen different percentages, have conducted different numbers of surveys. But what we really care about is the data, right? So this is showing, by default, it's just showing the most recent year's worth of data. And this is showing that seasonal timing of caterpillars. And it's quite up and down. It's quite bumpy here. There was this initial early June peak, dropped to 0. And then we had a later peak, a few up and down bumps. But we can add all kinds of other comparisons to the same plot. So we can add other arthropod groups. I'm going to add caterpillars, but from a different year to compare. And you can see, oh, wait, 2021 was actually really bad for caterpillars, not just in terms of the seasonal timing, but the proportion of surveys that had caterpillars, etc. We can compare caterpillars from a totally different site, or maybe let's say beetles. We can switch up. Sure, let's put on Acadia National Park in 2020. Oh, they had some data, so you can see. Oh, this other site. Now this comparison doesn't make any sense, but I'm just illustrating. You can compare site to site, year to year, one group to another group. So very flexible ways to visualize these data. Similarly, you can visualize the types of bugs that we're seeing. You can visualize how that varies from site to site. So we could add another site on here. We could compare to Acadia National Park. Looks like Acadia National Park in this pink color had more caterpillars per survey than here in North Carolina. We could, instead of comparing sites, we could compare, how does the composition of different bug types vary by month or by plant species, as I showed you in the presentation earlier. So all of these visualizations, oh, and I showed you examples of the trends. We can again add additional trend lines, as I showed you. All of the data for these is completely downloadable. You can play with it in Excel or something like that if you like. Let's see here. Going to address well, actually, before I address these new questions in the chat, let me just share one final thing. So here are the various ways you can visualize caterpillars count data. Also, I think of use is our resources page. So this is where you can find all kinds of training materials, written instructions for, okay, how exactly, I've talked about it, but can I get some more complete written instructions about how to set up a new site, how to train a new manager, or show off what all of the capabilities of a manager and managing their site are, tips for training participants or for testing out the mobile app. We have various ID guides, things like that. For educators, some tips for starting up a site at your school. Here, I alluded to the fact that you can do this on paper data sheets if you don't have access to a mobile app. Here's the instructions for building a beat sheet, for testing out that app, or instructions specifically step by step for going through how to use the mobile app. We've got various I won't get into it right now, but other tools for going in and viewing the data itself and correcting errors if you come across any errors. or exploring those data visualization tools I just kind of briefly went over. And then lastly, I alluded to these various learning activities. So again, the majority of these are not ones that we have created, but they've been created by other folks that are conceptually or thematically related to the project. And so in most cases, we're kind of just linking to these other possibilities. Okay, so let me stop sharing here and take a look at all of the, I know there's been a bunch of questions posed here in the chat, so let's take a look. Right, so yeah, native plant species finder, that's a great resource, and very much it's the case that, and you'll get a sense for this if you participate in this project, that certain kinds of caterpillars you really only find on certain kinds of trees or families of trees, etc. And in many cases, we don't have complete records of host plant use by caterpillars. So this project actually, especially if you take that photo with our app, that allows us to get that species identification of the caterpillar potentially, and allows us to better understand those host plant relationships. Marjorie asks, what about shrubs and bushes? Yes, shrubs and bushes are okay. So it's any woody vegetation. And we are again, the project initiated thinking about it from the perspective of foliage gleaning birds, right? And so it's not meant to be done in like an ornamental garden on ornamental shrubs and bushes so much. Although you absolutely could do that, but that's not the intention of the project. But even in the forest, like in the North Carolina Botanical Garden, we have a lot of spice bush in the understory. So we sample spice bush. It's not a tree, it's a bush. That's fine. So as long as it's woody vegetation. So Dave's asking about tent caterpillars that are not at eye level. Yeah, so this project, your job is not to document all of the diversity in the forest around you. It's to do some standardized monitoring that allows you to better compare from season to season and from year to year and from tree species to tree species. For bugs that only occur at the top of the canopy, yes, you'll never see them in this project. That's okay. So this project, the intention of this project is not sort of the BioBlitz perspective so much. It's coming up with reliable estimates of what's around. And that's absolutely a limitation of the project, is that we can only describe what we have at ground level. For people interested in better characterizing or actually feeling confident that what we see at ground level is representative of what's going on higher up in the canopy, which is a totally reasonable question. One project that we have gotten involved in is monitoring caterpillar frass. So frass is basically the caterpillar poo that rains down as caterpillars are munching leaves, and what goes in must come out, and ultimately it falls down. And so we have these so there's actually instructions on the website for if you wanted to get involved with frass monitoring. But what we've found is that the seasonal timing of caterpillar frass from the canopy seems to match pretty well the seasonal timing of caterpillars that we observe down at ground level as well. That may not be true everywhere or in all places. So that's actually a really important validation step. If you have people and the bandwidth to do that comparison, then we absolutely encourage it. Rebecca is asking, is a visual survey preferred to a beat sheet in most cases? A beat sheet seems very disruptive. And what's the protocol for dealing with insects once the beat sheet survey is complete? Like I said, we honestly do not have a preferred survey method because we recognize that different sites are under different constraints, resource constraints, time constraints, audience constraints of who they're trying to engage and what's the best way to do that. A beat sheet, it's disruptive in the sense that insects are getting dislodged from the branch. That's true. As long as you're not doing it in a way that's actually damaging the leaves and twigs themselves, that's okay. But Rebecca brings up a great point, which is that for caterpillars in particular, which are really the most vulnerable and dependent group of arthropods you would come across that really are dependent on being on that branch, we absolutely put caterpillars back on the branch. But for many of the other groups, they're very mobile. I mean, flies, of course, but even spiders, leafhoppers, things like that. Many of those, they're capable of flying and dispersing themselves to get to where they need to go. So for most other groups, after you do the beat sheet, if you've put the caterpillars back, for the most part, you can kind of shake the bugs back out over the branch, and some of them will fall on it, some of them will fall into the ground, but most of them will be able to get where they prefer to be pretty easily. So Dave is asking, I see a local site. It only has data from 2018. Is there a way to reactivate that site, or is it better to create a new one? Oh, that's a great question. So yeah, so certainly some sites, so if anybody participates even just one time and submits data, they'll show up on our map. And we want that data to be explorable. So we don't want it to go away, even if the site may not be active. But if you live near a site that looks like it's no longer active any longer, let me share my screen one more time here. If you go to participate, first of all, so, well, you saw on the map, you can of course zoom in on the map to where you live, and you can see if there are any sites nearby. But if you want to communicate with those folks, if you go to participate, get started, this link, find an existing caterpillars count site near you, actually has contact info for any site that is designated to be a public site. That is, it's not just someone's backyard. And so you can, it's currently sorted by state. And so you can see, you can find a site, and you can potentially find the email address of the person who created that site, who's the site owner. So yeah, I would absolutely suggest that as a starting point. I think all else equal, it would be better to revive an existing site, because that way you have already got this previous point in time. So now you have something to compare to from 4 years ago. And that would be great in and of itself, just to ask how much has changed in 4 years, right? If the person doesn't respond, maybe it was ephemeral, or the staff person has moved on, then yeah, absolutely feel free to create your own site. But I would say it's definitely worth trying to figure out whether there's anyone who might still be able to revive that previous site. And then Christy asks about specific dates to begin and end. And so you can do this project as long as there are leaves on trees. So that'll, of course, depend on where you are. Someone I saw is up in Minnesota with snow. Sorry. They're not going to be able to start the project till much later in the year than someone down in Georgia or Alabama, right? And likewise, they probably wouldn't be able to go as late into the fall as someone in Georgia or Alabama. So if you can, so we have a number of sites that really try to hit that full span of as long as there are leaves on trees, to give that full sense of that seasonal variation and fluctuations in timing. That's great. If you know that you have limited capacity for the volunteer base that you're tapping into, or you yourself are only available for X number of weeks, then the priority weeks, I would say, are really May through July. That's the data that's going to be most helpful when we relate it to trends in birds and bird migration and the timing of bird migration and bird reproduction. But also, as I've mentioned before, you might just do this on an annual basis. Maybe you want to just do an annual bug count, right? You can set up a bunch of branches, and you have a volunteer citizen science day or something at your site, and you get all these people out to do a bunch of branches, but you only do it once a year. If that's what you want to do, that's fine too. So of course, so from the scientist perspective, so having created this project, and I've been involved in analyzing the various data, depending on the question we ask, we will if we're asking a question about seasonal timing, and there are some sites that don't really have good seasonal coverage, they just have data from 2 or 3 time points, well, we'll just remove those sites from that particular analysis, right? We might not use your data if you didn't actually do weekly surveys for a couple months. That doesn't mean you should be discouraged from doing the project just because you only have the capacity to do a few dates worth of surveys. Like, if you're going to get something out of participating, regardless of how few the number of branches or how infrequent, you should participate, because it can be fun, and it can be a great learning experience, I think, to participate even at those lower levels of participation. And those data are still useful for other types of scientific questions. So questions, for example, comparing how many bugs are on this tree species versus that tree species, or in different urbanization contexts, right? In a highly urbanized versus more suburban or rural landscape, et cetera. So there's plenty of questions we can address with even just that minimal level of participation. So we are at 2:30. I am happy to stick around and answer any additional questions you all have. But hopefully you've got that link. You've seen the website. I've given you a flavor of the variety of resources that are available on it. If you have any questions, feel free to email us at caterpillarscount@gmail.com. But yeah, I would love it if we got some new takers from this webinar, and people were interested in learning more about the insects and arthropods in natural areas near them. So, like I said, happy to take more questions either in the chat, or you can just speak up at the end here. But everyone else should feel free to drop off. And like I said, I will try to email around a link once this is once this recording is posted. Great, thank you. Great to hear, Nan. And yeah, feel free to email if you have questions. As far as the FRAS survey methodology, yes, let me see what I have here. Here, for those remaining, I'll just reshare. I think this is in our For Educators page. Here we have a link. So here, this is actually a variety of side projects that you could do that are related to the actual bug monitoring itself. So here's a link to our FRAS fall monitoring project here. And FRAS fall, basically we use milk jugs, where you cut the bottom off a milk jug and you put it out with stakes in the ground, and you line it with some mesh. So there's instructions for how to set that up. And then you can sort of on a maybe every couple of days or a weekly basis, collect the frass, pick out the leaves and sticks and debris, and then drop that frass into just a little paper Dixie cup and take a photo. So that's the basis of our frass protocol. All right. Well, if there are no further questions, thank you everyone so much for participating, or in the webinar. And I hope to see some new Caterpillars Count accounts cropping up in the next couple weeks.